All right, so we're investigating in this lab the ability of plants, vascular plants in particular, to transport water with no apparent expenditure of energy. So what we want to do is confirm that the source for that of the force that moves the water is located where? In the leaf, exactly. Okay, we want to confirm, okay, that it is what we think it is, which is the osmotic pressure that is set up as water evaporates out of the stomates, okay, leaving salts behind. We want to confirm that that is in fact the source for the force that moves water through the plant. So in order to do that, we need to modify or manipulate the plant a little bit. Okay, here's how we're going to do that. We are going to take these plants here. Okay, these are called Dieffenbachia. I don't know why, but that's what they're called. Okay, and we are going to cut pieces of them off so that they are not connected to the stem for the most part. There'll be a little bit of stem, okay, or the roots. Okay, by doing so, will that eliminate those two parts of the plant as a source of pressure or force that could move water? Okay, so the next part of this is how are we going to tell whether water actually moves through the plant? Exactly. We're going to dye the water, and we're going to actually follow the path of the water through these leaves. Okay, the nice thing about Dieffenbachia is they have these very light-colored center parts to the leaf, and so dye shows up very well as it moves through the various parts of the plant that we've identified. First, the veins, okay, xylem, phloem, all that kind of stuff. Okay, so we'll be able to actually track it there, and oftentimes we can see where the stomates are on the bottom of the leaf. Okay, because as the water moves into those spots and evaporates, the dye gets left behind. And we can actually see the little dots that indicate where the stomates are. All right, so that's kind of what we'll be looking for in this lab, or what we'll be observing, watching for, etc. Okay, now, things that we need to keep the same throughout the lab, as best we can anyway, all right, would be what? What are some things that affect the growth of a plant or its ability to transport water? Right. We're going to make sure it's exposed to at least some sunlight. Okay. I mean, we can't say exactly how much. I mean, it's, late October, and we know that there's going to be, probably about 10 hours, if we're lucky, okay, of sunshine on these plants if it's a sunny day. But they're all going to get the same amount because they're all going to be in the same window. Okay. Now, if they're all in the same room, there's probably a few other factors that will be the same for all of them. Those you can figure out, okay, and write them down as your controlled variables. Okay. Now, I didn't outright say what the other two were, but I did hint at them quite, well, not obviously, but I hinted at them a lot, okay, for the other two, okay, for our manipulated and responding variables, that is. Okay, so for our hypothesis then, okay, for our hypothesis, again, we want an if, and then statement, okay? The if part should talk about how plants transport water. Okay, because that's what we're investigating. Okay, where's the force for the transportation coming from? Okay, where is it? Probably what is it? Okay, that stuff should probably go in the if part. The and part, as always, describes the experiment, and the then part should talk about what we'll see. And we just talked about that. We're dyeing the water. Okay, what are we going to see as a result? We're also going to be measuring the amount of water. So there's actually two responding variables in this lab. Okay, we are going to put these plants in a graduated cylinder. Okay, that is a cylinder with marks on the side of it that we can use to measure the amount of water. All right, so we will record how much water we start with, and then on Monday, we're going to record how much water is left. And so we'll know how much water has gone through the plant, as well as we'll see where the water has gone in the plant because the water is dyed. Okay, so there's actually two things that we're watching for, okay, as responding variables in this lab. Okay, and that's what you should talk about in the then part. All right, so the procedure we have to follow very carefully. Okay, so you're going to need to listen to these instructions and follow them to the letter. Otherwise, we'll have problems with our results, and that's going to affect how this all works. Okay. So what we're going to do is we're going to, first off, have to have two different graduated cylinders to start this lab. Okay, you're going to need one small one. Okay, I think they're 25 mils. Okay, those will be lots for what we're doing. Okay, so you'll start off with a 25 mil one, and you'll have a large one, 100 mil graduated cylinder. Reason for this is we need to mix the water and dye in the larger one, and then pour it into the smaller one. Because when we pour it into the smaller one, we're going to pour past the markings. The markings only go up to about here, right? We're going to need to put water up to about here because we don't want the end of the leaf to get out of the water over the course of the weekend, because if it does that, then it's going to look wilted on Monday, and that's not going to be good for our results. Okay, so we're going to want to fill it not right to the top, but very close to the top, so that there's enough room that when we put the leaf in, right, the water will rise as we displace water, that whole Archimedes thing. Okay, and we don't want it to come out the top. Everybody kind of follow me there? But the reason we have to have this cylinder is we won't be able to measure how much water goes into this second cylinder because we'll be past the markings. Right, so what we'll do is we'll put exactly 100 mils worth of water in the 100 mil cylinder, mix it, pour it in here, and then see how much is left in the big cylinder, and we'll know whatever isn't in there is now in the other one. Everyone kind of follow me on that? Okay, so that's what we'll do. That's why we have to use the big cylinder as well as the small one. All right, so we'll mix the water up, okay, and what color of food coloring should we use? Okay, we can use blue, we can use red. What shouldn't we use? Good. Yellow. Yeah, green and yellow are no good. Okay, and I'm just putting that out there because I had groups use them before, and it still boggles my mind that people did that. Okay, and then they came to me on Monday and they're like, Mr. Coderre, our lab didn't work, and I'm like, yep, you're right, it didn't work at all. And I wanted to say, because he's stupid, but I'm not allowed to say stuff like that. But yeah, it was pretty dumb. Okay, don't use yellow or green. I didn't even put them out. So you won't even have the option, because after that I just stopped putting them out. It wasn't funny, it was tragic. Okay. All right, so we're going to mix the blue food coloring in. You're only going to need like 4 drops max, okay, of food coloring in 100 mils of water. You put any more in there and it's going to be like soupy. Okay, we don't need it to be that thick with the food coloring. 4 drops of food coloring will be less. You can even mix the red and the blue if you want, and you can have purple. Okay, we got all sorts of freedom of expression in science. Okay, whatever colors you like except green and yellow. All right, so you're going to mix the water first. Okay, then once you have the water mixed, actually you're not going to do this step yet. Okay, when you have the water ready, the graduated cylinder ready, then you're going to come up to the front, and I'm going to give you the finger off of a rubber glove. You all are thinking something else, okay? All right, so I'm going to give you that, and you're going to take it back, and you're going to put it over the top of your graduated cylinder. Why are we putting a rubber glove over that? Yeah, we want to limit the amount of evaporation that occurs out of the cylinder. We want most of the evaporation to be out of the leaf. So we're going to put a finger off of a rubber glove over top of it, poke a hole, a small hole, like with your pencil, in the rubber glove, and then you'll come up and get your plant, okay? So when you get your plant, it is important that you follow these instructions very carefully, okay? I'm going to have these plants submerged in the big sink at the front, okay? You need to cut your piece of plant off under water. And before you take that piece of plant out, you need to put your thumb over the cut end. Why? Not to keep water from going into it, to keep, and keep air from getting in the xylem. If air gets in the xylem, it will disrupt the whole polar nature of water, cohesion, adhesion thing that's going on inside the xylem tubes, and we don't want that, okay? Because if it happens too much, then water won't be transported very well in our plant. So you're going to have your thumb over top of it, okay? Pick it up, and you can see that in, I think it was this picture here, okay? She didn't do it, okay? See her thumb? It's there, okay? This was just to demonstrate. This was actually a piece that had gotten cut off and no one had used. But, okay, this is a lot more leaf than you're going to need, Okay, if you get like one really big leaf, that'll be good. Okay, or a couple of smaller leaves. We want to try and cut them from the same piece of stem. Like if they're attached, that would be best. Okay, if they're not attached, well, then we have to deal with it. But hopefully you can get either one very large leaf, okay, or two kind of medium-sized leaves. Right, we've got 2 plants here, so there's lots of leaves for our group sizes, okay, in here. Everybody with me on that? So cut them under the water, put your thumb over the end, okay? Then you're gonna go back and you're going to put it, okay? You're gonna very quickly put it into the cylinder, okay? Push it down so that the stem is well into the water so that over the course of the weekend, it won't evaporate enough water to expose the bottom, okay? Everybody got it? All right, once that's done, okay, I'll give you a piece of tape and you can write your names on it, put it around your graduated cylinder, right? So that you'll know you're planting on Monday. The observations you need to make today of your plant are, you need to have a record of the amount of water, okay, that's in the cylinder today. That'll be your initial volume. And you need to take a variety of pictures of your plant today. Those will be your before pictures. All right, so I would suggest that when you are taking pictures of your piece of plant, you take a picture of the top of the leaf and the bottom of the leaf, okay? Make sure you get them in focus. And as much as you can, the stems, and then probably one of the whole apparatus. Okay, so like that. Then you're good. That's the observations that we need to make today. On Monday, we'll go back in, and all we will do is, again, we'll have a look at our leaves. We'll take after pictures of the leaves and plant, so take exactly the same pictures you take today. And then we'll take the plant out, and we'll measure the amount of water that's left in the tube. So we'll have to pour it back into the 100 ml cylinder and record how much is in or was left in our smaller cylinder. Okay, and that'll be it. Then we'll do the analysis and stuff after that. Okay, so questions on what we're doing? All right, so we'll go across the hall, we'll get it all set up, and then we'll come back and I'll explain the analysis and stuff that we'll do on Monday. All right, so now that we have those apparatus all set up, okay, they will run over the weekend, okay, and they will transport water into the leaves. Everyone got their pictures? Okay, so on Monday then we'll take the same pictures and we'll measure the water and we'll have our observations, and then you guys will be able to do your analysis. Now, in terms of your observations, what I am looking for is your before and after pictures, okay? So you'll need to include those, insert them into the Google Doc, okay, as well as your initial and final volumes, okay? Because you're going to be referring back to those as you answer these questions. Like question number 1 is really easy. Calculate the amount of water taken up by the plant, okay? So you take your initial, you subtract your final, boom, you got that. Okay, that's pretty easy. Explain any differences between the initial and final values for the volume of water. You don't have to worry about mass of the setup. That's not part of it anymore. Okay, compare the rate of transpiration in your experiment with that of other groups and explain any differences. Right, so you want to have a look at what other people's numbers were, and then obviously have a look at their plants, see if there's any reason for why yours might have transported more or less than theirs. Second question, what evidence do you have that water followed the path discussed in class? Explain. So again, you're referring back to your observations here, okay, looking at where the dye is, all of that kind of stuff. And third question, predict the effect of adding salt to the water. Okay, describe how you could test this prediction. So what would happen if instead of using regular tap water, we used salt water instead? Okay, how would the experiment go differently? What kind of observations do you think you would get and why? Right, or what we are looking for there. Then for conclusion, copy and paste your hypothesis, explain why you can accept or reject it based on your analysis and observations. And 2 sources of error for the experiment, explain how you could eliminate them from future labs. And then the class discussion, obviously you don't need to do those. We'll talk about those when I hand back the labs. All right, any questions on the expectations for this? All right, so you have the remainder of class to work on this, or maybe your cell transport activity, okay, that we started the other day, whichever. You've got the Chromebooks for the remainder of class, so make good use of the time.